Hello, welcome to Saturday Morning NeuroSim. We haven't had a meeting in a couple weeks, and I wanted to have an update, go through kind of a normal meeting with readings. We're not going to have any discussion. I'm going to be the only one here, but going through different features like we would in a regular meeting. So the first thing I wanted to give is an update. So our Google Summer of Code scholars are hard at work. They're working on their projects. We're now in week 7 of the program and several weeks into the coding period, and they've been turning out great code. We have Vidi, who's working on the open source sustainability project, and she's been populating her repository with her project that involves agent-based modeling and reinforcement learning. She's using the SARSA approach and, coming up with some really good metrics for measuring a lot of this. And so this is, coming along pretty quickly. I'm very impressed. Lalith from the DevoWorm group is working on DevoGraph, and he's also been pushing his code to the repository. And he's been working on hypergraphs, specifically different formulations of hypergraphs, building visualizations of hypergraphs, and mapping that to the biology, which I think is also very important. Our other GSoC scholar, also involved in DevoWorm's DevoGraph project, is Jayadratha. Jayadratha has been graduating from his graduate program, so he's been a little behind our other two scholars, but he's still working on this model of temporal hypergraphs. And so he's been also contributing to the repository. He's been building visualizations, nice visualizations of these temporal hypergraphs and the temporal structure of development in particular. And him and Lilith have been working on different aspects of the biology. So this is going to be a nice addition to DevoWorm as a group, but also the DevoGraph project. So that's what our Google Summer of Code scholars have been up to. This week we had 2 meetings. The first was the DevoWorm meeting. We usually have that on Mondays. And we talked about a number of topics. We picked some papers from our collaborators, one on muscle cell growth, and we also did some things on developmental mechanics. I can link those together. And then we had a discussion on some research that Lalith is doing with Richard Gordon on syncytium and C. elegans, working out the lineage tree for the syncytium. Lineage tree typically bifurcates, but in the case of syncytium or fused cells that form a tissue with multiple nuclei, they reticulate, they merge back together. A lot of these cells fuse, they come from different parts of the lineage tree, and they fuse together into a phenotypic structure. So we talked about that. I also talked about this article on lifelike simulations. So lifelike simulations are lifelike simulations. They look like life, they act like life, but they're not living systems. And more importantly, you don't need to use a representation of any known living system to achieve them. So if we think about systems like morphogenesis, or systems like ant colonies, or systems like bird flocks, those all have agents or some sort of biochemical set of reactions, behaviors generated by brains, etc., etc. They have some biological substrate to them, or biochemical substrate. In the case of lifelike simulations, you can simulate processes that look like those systems have a substrate, but you don't have to model the substrate. You can model, in fact, with any generic mathematical model. And this is the logic that Stephen Wolfram uses to make statements about nature using cellular automata. In this case, they were working on a agent-based simulation that looked like life. And it's really hard to say what life looks like. You know when you see it. You can define different systems like emergent systems, percolating systems, systems that incorporate migration of particles or the organization of particles, spontaneous organization, things like that. And so usually we have a set of conditions for lifelike simulations, and I talk about that in the meeting. Okay, the other meeting we had was on Friday. That was the open source meeting that we usually have. We had our Google Summer Code updates, and then we had a nice discussion by Morgan on the things he's been involved with bioprinting, organoids, and building tissues and doing tissue culture, and ending up with something approximating a customized organoid with a customized form and a customized biology. And so he's been going to workshops, going to online workshops, in-person workshops, and exploring this concept with a number of labs and trying to figure out the way forward on this. It's really exciting work. And then Jesse, of course, he's involved in, he's doing this micro MBA, and he's doing some interesting things with organizational research and project management, which is one of his specialty areas. And then myself, I presented on something called open washing, which is where people present projects as open source, but they're not actually open source, not at least in the conventional sense that, say, like a free and open source software or a FOSS organization would present it. And so this was an interesting discussion on how materials are open, how they're transparent, the licensing terms by which they're released, and of course the organization itself. What are their goals, and what are their, how do they envision these materials being used versus how do the contributors do it? So there are a lot of opportunities there, but also a lot of points to be made for not being totally honest about those, aligning those goals. Okay, so that's it for this week. In terms of updates. Oh, hi, how are you?
I'm good. Sorry. I lost track of time trying to build CompuSoul 3D.
Oh,
it's
okay.
their Python day. They're gonna do their summer school like end of July.
Oh yeah.
Beginning of August. So, move to it. So anyway, yeah. So I'm in Roanoke, Virginia.
Oh yeah. That's great. So you were at the workshop, or you?
I was left like late Thursday night, and came back here because my younger kids are here with my brother's kids, so it's cousin camp.
Yeah.
my parents, left
right.
my parents.
That's good.
But really, really remarkable. Like this is a way to build microfluidic systems,
right,
with benefits.
Right.
Like so it's like you one, the equipment is inexpensive.
Mm-hmm.
Right? So it's just like it's a modification of a standard consumer 3D printer.
Yeah.
Two, you print in a bath. So that's what they're calling fresh printing, and it's free form, reversible, something.
Yeah.
But the point being that like you're choosing a hydrogel where when you extrude into it keeps that shape. So it's just like the hydrogel moves for whatever you've extruded, but then it keeps that material. So it's just like as you're printing in this kind of semi-solid bath, right? Like you don't have to rely on gravity, or basically gravity doesn't hold in this bath. So you can print in 3D by moving your syringe point to wherever you want, right?
Yeah.
So you go through and you can print these super accurate, super intricate shapes in this gel, and it holds that form, right?
Yeah.
And then you can later heat up then you put it in an incubator, and the gel melts, right? And then it's just like, and then you get whatever you printed.
Right.
I mean, and then maybe you've got to do some extra washes and things like that. But then what's cool? So we did the this was just tests. It was really about the printer conversion, in some degree, like the fresh printing. By printing in this gel. The first bioink was this alginate, and it was really just to print something else, something other than what we into the bath. But then the second stage is using what they call chips, which is this collagen-based perfusible scaffold. And so now imagine where, like you've seen these kind of microfluidic chips. And there's a couple examples that I think are maybe good to look at about just microfluidics, but one of those is using microfluidics for a mixing process. So we've got two inlets, separate, what will be separate materials, and then they come together, and then you say maybe snake back and forth, what they call a serpentine pattern, and then one outlet. So people will use this both to model physiological channels, but also just by putting in two things, they're actually doing mixing. and one example of that is actually lipid nanoparticle fabrication. So you can have DNA and lipids and get encapsulation in the mixing, just as an example. But in this case, the main thing is that, like, instead of doing kind of clean room fabrication of these microfluidic chips, you're doing fresh printing. So you're doing this printing in a bath using collagen. You can print something that looks, in gel, it looks plasticky, in the sense of like it's super well defined. But now, if you melt it, I mean, even when it's just melted and still kind of in the gel solution, you can see when you just tap it that the collagen is super soft and super. I guess the technical term would be fluffy. I don't know what but really interesting. because you can still both use it in that kind of microfluidic way, right? In the sense of like there is an extra step in terms of just how do you interface with tubes, right? Because it's so soft material, you can't just jam a plastic pipe into these things.
Mm-hmm.
So there's an extra piece to make for the two inlets and outlets in terms of how to interface with your peristaltic pumps, right? But again, this is kind of a feature, not a bug, because what that allows you to do is again the softness is because you've printed collagen, and so you've got a biomaterial that you can either seed. I mean, so you can actually create a cell bioink that prints with the collagen, right? So in that case, like your material already contains cells, or you can perfuse cells through this microfluidic collagen platform,
yeah,
and get that kind of wall-based, like covering the channels and things like that.
right.
So again, like one of the I would love it. I mean, I get why they're saying like we're doing, they're almost like we're doing this as a service to the open source community. Like we're teaching this thing, because I was just like, you need to do like a 2-day workshop on microfluidic platforms, and they're like, I think you misunderstand why we're doing this. like we don't have a grant to teach this.
Right.
I think was kind of their point.
Yeah.
And but that's okay. That to me is just an opportunity, and that because I said, this is I think a really undersold, or I didn't appreciate it until I got there and saw their applications of it, as well as like hearing people talking about it, where it's just like there are actual limitations to the current fabrication technologies, in that you are making these fluidic platforms from these kind of artificial materials, specifically because most of them are used for drug studies,
Right.
and they don't want the drugs to seep into the walls.
Oh, okay.
So like they don't, they're kind of like artificial to preserve, to make the system simpler, but it also makes them less realistic,
Right.
or less accurate, or and so it was super interesting. I mean, again, like I'd love more expert backgrounds in microfluidics. I mean, the other funny thing showing up at this thing was coming with two, like one, we were biopump lab, so everybody was just like, you're who? And yeah, so trying to explain to them what a community science lab is.
Yeah.
But the other was that, like, that everybody else showing up, like, either sort of had a connection to this lab. It was basically, this was a workshop that they were doing for their collaborators.
Oh, okay.
So it was just like, it was like a lot of groups from Pittsburgh that are like from neurosurgery or from other, other departments outside of BioE, right? So it's just like, it's like these guys are BioE. The other groups were like the applications people from dermatology, pulmonary, liver, like all these different, super interesting one that was focused on cilia, and anyway, it's nobody was doing neurons.
Yeah.
Except, the one group that looks, it's coming from a kind of neural translational engineering lab, definitely had a lot of experience with culturing neurons, a lot of experience doing rat surgeries, things like that. And they were just like, neuronal culture is the hardest. and but I think they understood that, like, if you can get that to work, that you really have a super interesting opportunity in terms of the kind of constructs that are possible with this. So the way in which, the cultures will be fundamentally different from 2D neural culture. And you can, it's very new territory. Is the other interesting thing. And certainly the BioE, the CMU, PIT, BioE groups that did this certainly see how this can play into kind of like shaping organoid culture, potentially too. And just very, very exciting.
Yeah. Well, that sounds good. Yeah. I'm glad to hear a little bit more about that. Yeah. And I don't know. I guess it's very experimental. It sounds like it's not like they've really kind of gotten outside of their local experimentation and stuff like that. But yeah, it sounds.
But they're like at the same time, they're far along with muscle, with cardiac tissue. They're far along with epidermis, pancreas, like bone plus muscle plus skin. Vessels, both blood vessels and heart pumps, heart valves. There's definitely an emphasis on kind of like muscle skeletal, as they point out, like this, it's the largest organ in the body.
Yeah.
So there's kind of a reason for that. It's definitely partly a kind of like an ease. It's like it is, easier. It's not easy, but it's it has certain things that they can. Again, like muscle just being these things where they can almost print protein, and so they're far along, and there's some other interesting. Anyway, it was also great because they could really give us a breakdown of like who's doing anything else.
Yeah.
And connecting us with some great people in the Bay Area too.
Okay.
So that's exciting. And there's groups there both doing like printer development with some other kind of features that are interesting and accessible for the kind of thing we can do at the tower, as well as like the Center for Cellular Construction that's like definitely pushing every single kind of synthetic biology direction. So yeah, it's experimental, but like outside of the brain, there's a lot of activity and a lot of like successes happening. Like people getting lab on a chip stuff done.
Right.
And like I think we've talked about before, like lab on a chip seems to be the way to do, the way that people are doing new stuff.
Yeah.
And and we said there was a lot of joking about how with the crumbling American democracy, there is a funding opportunity for alternatives to animal testing.
Yeah.
I'm not saying it's a silver lining.
Right.
But these are people who are well positioned for if there's a demand for like at least an alternative plan for animal testing, like these are the kind of groups or this is the kind of technology we use.
I like that how this combines 3D printing with cell culture with tissue engineering, and then gives you that soft material kind of platform to work with. Yeah.
So like then just coming off the back of that, and doing some research on the way home, it's like it's no surprise that some of the best kind of similar work that I saw in musculoskeletal bioprinting was coming from like soft robotics lab at ETH Zurich.
Okay.
So definitely, there's a lot of overlap there, and I love that Feinberg just came from. He said the most interesting conference that he's been to recently was the biohybrid. It was like biohybrid robotics meeting, and that's where he met Michael Levin. Yeah. And when he saw, this is the other cool thing, is that he saw the Anthrobot stuff, and he was just like, hey, wait, this is very similar to what we're doing with 3D printing and cell culture. And so like because they're a kind of deep cell culture group, like they already work with embryos for other reasons, and so he and I ended up talking late at dinner, and like how kind of like the Anthrobots are built, and like how accessible that equipment really is, and so yeah, all the complexity now lies with the cell culture work.
Right.
I mean, it's just like you still have to nail the cell culture work.
Right.
That's the small pipe. That's also the expensive pipe. or
Yeah.
like the expensive stuff. Talking to the Biopit group, that really was like the brains behind the 3D printer kind of jailbreaking and conversion. Like his lab costs. I mean, so he confirmed that like his biggest lab setup cost was microscopes,
Right.
and his biggest recurring cost is cell culture reagents.
Right.
It's just like that's after people, that's what we spend the most money. Which again, these aren't surprises, but I've just been trying to get that across at the lab in San Francisco. That like we need to fundraise for microscopes because it's like it's just really hard to do for less than like $100K.
Right.
And we've got to finish our BSL2, which even when complete doesn't mean that you can actually nail the kind of cell culture that you want to.
Right.
And the stem cells for neuronal differentiation are kind of like again the most notoriously hard ones to tackle. But that just one final thing just was really interesting and exciting in terms of also how a lot of these groups are integrating both synthetic biology and genetic modifications. The keynote that they brought in from the outside from Penn State. Obviously, the other thing is that almost all these guys also have companies. So this is like they all certainly see. I mean, the organoid people are just as bad.
Right.
So I get it. Like they all see a commercial future for this.
Right.
Or they all see it to some degree. I can't blame any academic for thinking that they should be starting a company these days, just because of funding. But but I think they all see the commercial potential, and feel like they should be taking out patents and getting a commercial entity lined up to be a vehicle for this.
Right.
Yeah. So diving now into IPSC kinds of helpful protocols and techniques to make it potentially doable in a low resource community biolab space.
Right.
But feeling really good about the printing and what that, kind of like the features that offers.
Oh, great. Yeah, that's great. Yeah, I wanted to go over some papers and things like that. They're kind of that's a tangential turn from like what we're talking about here, but I have some interesting things in the queue we haven't gotten together in a while. So actually, this paper is maybe somewhat related. It's not really that related, but it's something new it's a review by Adam Safran, Ines Hippolito, and Andy Clark. And this is Frontiers in Neurorobotics. And this is it's an editorial, I think it's a special issue, BioAI from embodied cognition to enactive robots. So, this is where they're thinking about enactivism, embodiment, intelligence, dynamical systems, robotics, and putting all that together in kind of a theoretical framework. So it's not, this kind of bio-development of robots. It's this sort of theoretical aspect of it. So why don't we go through this? So there's an introduction here where there's an Emily Dickinson poem talking about the brain, and then they start in, if the connections of the human brain were disentangled and placed into a sequence, they would indeed be wider than the sky, being hundreds of kilometers long and likely capable of stretching to the moon and back. So they reference wider than the sky in this poem. If we consider the kinds of intelligence generated by brain-body environment systems, then such emergent minds may be vaster still in terms of their complex combinatorics, with the pinnacle of expressive power potentially being found in language with its infinite use of finite means. And so this is just kind of the sort of generativity of language. That's what this refers to. The field of artificial intelligence and machine learning, AI/ML, seeks to reproduce the powers of biological learners, where we struggle to recapitulate the ways in which even supposedly simple animals demonstrate the ability to respond flexibly to a wide range of situations. In this research topic, we were grateful to receive a diverse assortment of articles that address ways in which principles of enactivism and embodied cognition might allow for advances in AI/ML, potentially without requiring explicit representations, pre-specified algorithms, or centralized control structures. In what follows, we briefly summarize these contributions, highlight some potential implications, and end with the discussion of potential ways forward for AI/ML and cognitive science more generally. So there are a whole bunch of papers in this collection. I think I remember seeing a solicitation for this, and it may have grown out of a collection from the Embodied Intelligence Workshop, I think about 2 years ago. I think they were soliciting for this issue and then another issue at the same time. So this was, I think, the one that they solicited, that is Frontiers in Neurorobotics. And I think we decided not to do that because of the cost of publishing an article. So this is, so they kind of go through the different articles in the special issue. So the first one is the acquisition of culturally patterned attention styles under active inference. That's by Axel Constant et al. So if we go to that paper, here's this paper here. Andy Clark is on this paper, Axel Constant, and some other people. So this is an active inference-based simulation study of visual foraging. The goal of the simulation is to show the effect of the acquisition of culturally patterned attention styles on cognitive task performance under active inference. We show cultural artifacts like antique vase decorations drive cognitive functions such as perception, action, and learning, as well as task performance in a simple visual discrimination task. We then describe a new active inference-based research pipeline that future work may employ to inquire on deep guiding principles determining the manner in which material culture drives human thought by building and rebuilding our patterns of attention. It's a pretty interesting paper. So that's the first one. The second one is enacting plant-inspired robotics. This is Leanne Calvo. And this is where they look at plants as a holistic source of inspiration for soft robotics in terms of their non-centralized modular architectures and highly plastic phenotypes. This was something that I saw actually at the Embodied Intelligence Workshop like 2 years ago. They were showing their work, or they were building. It was basically something they called robots, which you see here. So this is where you basically build an agent that acquires sources of matter and energy and puts them into growth or growing a phenotype. And so they have this whole area that they're kind of pioneering called robots. And then they get into what happens when you're growing your phenotype in situ. So you have so in contrast with notions of autonomy based on the independent operability of systems over an observation window, Plants and other living organisms exhibit a stronger form of autonomous functioning in terms of needing to support self-production dynamics that create distinctions between themselves and the domain of interactions that maintain the conditions of viability for the system. So that's work on plants, but it's also that kind of work where you want to grow a robotic system, and you want to have a system that can be plastic, can have these modular architectures, very different from sort of an animal-inspired or in a lot of cases mammalian-inspired robotic system. This is another one, carving up participation, sense making, and sociomorphing for artificial minds. This is on social cognition and participatory sense making. This one, embodied object representation, learning, and recognition, shows how robotics can be informed by considering the ways in which biological agents achieve scene understanding or adaptive object manipulation and navigation capabilities by leveraging active interactions with the world in their first encounters with novel situations. This is inspired by neuroscience theories in which neocortical columns build predictive models about objects within allocentric reference frames. And this is what they call the cortical column network architecture, or CCN architecture. So I don't think this is the group, the Numenta group. I think this is a different group, but they're kind of focusing on the same idea.
Yeah, I mean, I was definitely, in my head, I was just like, that does sound like Thousand Brains.
Yeah.
But who's was it Van de Maele?
Van de Maele. So this paper has, Toon Van de Maele.
Oh, Tim
Verbalin, yeah.
Okay.
So,
but Verbalin's at CMU, right? So
at me, yeah.
So it's like a bunch of Ghent people plus Tim?
Yeah. I see there's someone from Numenta who reviewed it. Supatai.
There you go.
Yeah, well, I guess it was good enough for them, so I guess.
So reviewed by Carl Friston and Supatai.
Yeah.
Okay, that's funny. Yeah, okay.
Yeah, and again, this is on these kind of cortical column networks, and it's based on the visual system. So you're trying to again do visual perception, and this is pattern recognition, just in a little bit different way than we're used to with neural networks. Yeah, so that, and then there's this next paper, Grounding Context in Embodied Cognitive Robotics. This describes how autonomous machines may be augmented with greater behavioral flexibility by providing systems with a global context that integrates agent-related environmental and task-related information. Through the interaction of these core elements, agents are capable of three things. The first is selecting self-relevant tasks on the basis of current and anticipated future needs. These could be for, say, like learning or mastering contingencies. The second is performing tasks with a continuous performance monitoring. And then three, abandoning unsuccessful tasks based on overall prediction errors during situated action cycles. So this is, the kind of how they think about grounding, which is a classic theme in cognitive science. And so that's this paper. And then the next paper is The Problem of Meaning, the Free Energy Principle and Artificial Agency. This is Julius Kiverstein et al. Describe how biological agents solve the problem of meaning by acting in ways that express sensitivity to context-dependent relevance. So this is very similar in term, but this is cast in an enactivist framework. The authors argue that robustly autonomous agents require stable, self-sustaining patterns of sensorimotor interaction to ground values, norms, and goals as they encounter different and differently meaningful environments. So they talk about enactivism as well as the free energy principle, including the challenge that these perspectives are fundamentally incompatible with biological systems exhibiting historical path-dependent learning, but with free energy minimizing agents severing this historicity. So free energy minimization is where you're selecting from an ensemble, and then you're optimizing for the, the minimal or maximum, as opposed to biological systems where you have this historical path dependence, meaning that the thing that came before influences the thing that comes next. So it's, free energy principle, you could think of it as sort of being ergodic or something like that versus like historically contingent. Such free energy principle agents also show a lack of interactional asymmetry present in enactivist accounts of autonomy. In these fundamental, in addition to addressing these challenges, it is suggested that rather than fundamental incompatibility, the free energy principle needs enactivism for the problem of meaning, and enactivism needs the free energy principle for precise formal modeling the necessary constituent factors for realizing agency. So a lot of stuff there on enactivism and the free energy principle, perhaps reconciling them, and then reconciling that with cognitive science, and seeing where the sort of the connections are there. Okay, so the next paper is Avoiding Catastrophe: Active Dendrites Enable Multitask Learning in Dynamic Environments, and this is Iyer et al. This is where they introduce a neural network architecture for enhancing the embodied systems to operate in dynamic environments while flexibly adapting at changing task contexts and continuously learning without catastrophic forgetting/inference. So this is where they're trying to overcome or make the system robust to catastrophic forgetting. A lot of times you'll get this if you change tasks or task contexts. So you learn something in one context, you switch to a different context, and everything you've learned is now generalizable, and you have to relearn everything. This is, of course, if you have a generalizable model, this isn't as much of a concern, but people have been trying to figure out how to generalize from a training set for a long time. And so one way around that maybe is to build a system that's robust to catastrophe. And that's what they're doing here. This is achieved by incorporating active dendrites in sparsity promoting local inhibitory systems, so dynamically constraining and routing information in a context-specific manner. The architecture is tested on several benchmarks, including a multitask reinforcement learning environment, in which agents must solve a variety of manipulation tasks, such as meta-learning, in addition to a continual learning setup in which task predictions change over the course of training, which is reversal learning. So meta-learning versus reversal learning. In both simulations, the architecture developed overlapping yet distinct sparse subnetworks that mediated the fluid adaptation to multiple tasks with minimal forgetting, providing for the first time a demonstration of high performance with respect to both multitasking and continual learning. Okay, this actually sounds like maybe some of the sort of broad outlines of what we're trying to do in the Ever Good Regulator theorem paper. At least thinking in terms of some of the ways to think about learning and networks and regulation, at least in the EGRT paper regulation. They don't use regulation here. But yeah, it's like this idea of solving or potentially solving a lot of the problems of modern machine learning with some sort of newer paradigm, an experimental paradigm, where you can get different types of learning, different types of training regimens, And it gives you these different levels of robustness in the system. So the next paper is in Social NeuroAI. Social interaction is the dark matter of AI. Bolota and Dumas introduce a three-axis framework for social learning and biologically inspired AI. And I guess this is informed by the free energy principle. So there are three of these. The first is brain-inspired models of cognitive architectures, such as global workspace and attention, schema theories that bridge individual and social intelligence. Two are dynamical systems perspectives for handling the inherently time-dependent nature of cognition. And then three is embodiment as a source of sophisticated communicative signals. These social interactions are essential elements of advanced cognitive ability, yet remain underexplored in AI, constituting the dark matter with respect to attempts to understand humanimal, I guess, like intelligence, so basically animal-like intelligence. In light of this gap in our understanding, the authors review the role of social learning in cognitive development and this new field of social NeuroAI. Then there's this other paper here, goal-oriented behavior with a habit-based adaptive sensorimotor map network. And this goes through sort of this goal orientation and then habit and kind of going over how to implement a habit-based robot controller that draws on enactivist principles to realize agency by an adaptive sensorimotor map network architecture. This paper here, in Embodied Intelligence, Smooth Coping in the Learning Intelligent Decision Agent Cognitive Architecture. Cronstedt et al. Describe how skillful interactions may become habituated and ingrained through experience, thereby placing less stress on cognitive load relative to considered and deliberate thought and action. These are just things like walking, driving, skiing, short order cooking, etc. And then this gets into some philosophical literature, philosophical arguments, and then kind of thinking about this in terms of a theory. Looks like Piccinini, who we know from the discussions we had on the brain as a computer and things like that. He has a paper in here, Situated Neural Representations, Solving the Problems of Content, where he argues that situated approaches to mind based on embodiment, embedding, and action affect are deeply intertwined with neural representation, with such a computational approach requiring embodiment, embedding, and action, and affect at its very core. Situatedness is suggested to be necessary to describe the adaptive shaping of computations in a number of ways. This one here is an enactivist-inspired mathematical model of cognition. This is where they look at sensorimotor cognition and model this using deterministic automata. Then there's this paper, Using Enactive Robotics to Think Outside of the Problem-Solving Box, how sensorimotor contingencies constrain the forms of emergent autonomous habits. And then this is kind of interesting given what we were talking about earlier. This suggests that AI ought to take inspiration from the precarious, self-maintaining organization of living systems. They demonstrate how robots controlled by an iterative deformable sensorimotor medium, or DSM, can realize the spontaneous emergence of an organized ecology of habits, capable of reenacting adaptive behaviors, with habits formed within modalities having relatively greater similarity to habits across modalities. And this is similar to observations of biological systems. This talks, I'm going to look this paper up and open it up. And this is Okay, so let's look at the abstract. We suggest that the influence of biology and biologically inspired robotics can be embraced at a deeper level than is typical if we adopt an enactive approach that moves the focus of interest from how problems are solved to how problems emerge in the first place. In addition to being inspired by mechanisms found in natural systems or by evolutionary design principles directed at solving problems posited by the environment, we can take inspiration from the precarious, self-maintaining organization of living systems to investigate forms of cognition that are also precarious and self-maintaining, and that thus also, like life, have their own problems that must be addressed if they are to persist. So they use a simulation to explore precarious self-reinforcing sensorimotor habits as a building block for a robot's behavior. Our simulations of simple robots controlled by an iterative deformable sensorimotor medium, or IDSM, demonstrate the spontaneous emergence of different habits, the reenactment, and the organization of an ecology of habits within each agent. The form of the emergent habits is constrained by the sensory modality of the robot, such that habits formed under one modality, say vision, are more similar to each other than they are to habits formed under another, let's say audition. And so we discuss these results in the wider context of four things. The first are enactive approaches to life and mind. The second is sensorimotor contingency theory. Third is adaptationist versus structuralist explanations in biology. And then D, the limits of functionalist problem-solving approaches to artificial intelligence. So what we do now is functionalist, and what they're suggesting is that there's maybe the structuralist explanation. There's this, in biology, of course, we use an adaptationist explanation. So functionalist for modern AI systems, adaptationist for modern biological systems, and then they're talking about maybe more of a structural explanation. So that's kind of where they're going with this paper. Actually, they have a nice part in here on alternative conceptions of the mind. And this was sort of goes back to cybernetics. So they cite Grey-Walter and Ashby. And so these are, of course, early cyberneticists who are doing things with robots and with models. And so they say alternative conceptions of the mind were available at the origins of artificial intelligence and robotics, but the rapid success of computer science left them aside. Over time, the limitations of the problem-solving centered computational theory of the mind became apparent, and the biologically inspired, embodied, and later enactive conceptions of the mind gained momentum. We are now immersed in a mesh of hybrid architectures applied to a wide range of practices, from industrial to scientific modeling applications, and a new summer of artificial intelligence is rising, with robots as a major container of social and technological expectations. So they kind of cite the early cyberneticists, and they sort of say that there was this alternate path that was never taken. But I'm not sure that they actually explore back down that path. Nevertheless, I think this looks like a really good paper in terms of framing maybe some of the assumptions that we're making in our models. Then we have other papers. This one is Reach Space Analysis of Baseline Differential Extrinsic Plasticity Control, or DEP control. And this is where you're looking at sensorimotor or I guess in this case, goal-free simulated agents that produce environmentally aware behaviors. So they further extend this mechanism to intentional behavior to determine whether short-circuited DEP or differential extrinsic plasticity can generate desired trajectories in a robotic arm by a simple open-loop control with transient and limit cycle dynamics explored in experiments involving target reaching and circular motions. So this almost sounds like an alternative to control theory for robots, where in control theory you're often using feedback and you're using this sort of goal-oriented feedback mechanism and control plant mechanism to guide sensorimotor behavior. In this case, there are these goal-free simulated agents producing environmentally aware behaviors, meaning responding to their environment, and then building these open-loop systems that could perhaps generate similar trajectories. This next one is resonance as a design strategy for AI and social robots. Thinking about the physical mechanisms of resonance and adaptive resonance with human experience, with consideration for enhancing those experiences with human robot interactions. This one here, with self-concern across scales, a biologically inspired direction for embodied artificial intelligence. SIMS focuses on a foundation for intelligence for all biological systems that reflects an existential tasks of continued viability. So this is like that other paper we saw where they talked about, this was the Egbert and Barandiaran paper, where they talked about precarious self-maintaining organization of living systems. In this paper by SIMS, this is sort of this continued viability idea. Self-concern is introduced as a property of a complex system that describes its tendency to bring about states that are compatible with its continued self-maintenance, a potential means of recapitulating the power and principles of human-like intelligence in artificial systems. So there are a couple more papers here I'm not going to go over necessarily. I think this is another paper by Garner and Egbert. Embodiment enables non-predictive ways of coping with self-caused sensory stimuli. This demonstrates how sensory attenuation for self relative to externally caused stimuli can be explained inactively. So there are a couple more papers here I'm not going to go through. But I think you get the sense that there's a very heavy emphasis on embodiment and enactivism, and kind of framing a lot of traditional questions in cognitive science in that way, and looking at how those concepts fit into things like the free energy principle and sort of cognitive sciences. There's this sort of focus in cognitive science on problem solving and function. So usually if you're describing something like attention, there's this functional description of attention. Attention does something, it processes information, it gives you this output. It's an input-output scheme. And so that doesn't necessarily consider, other things that are going on with attention, maybe its origins or other things that might be of equal usefulness in applying this to, say, robotics or artificial intelligence or something like that. And I don't know how closely this hews to some of the like the Anthrobot stuff and the Biobot stuff, but there was a lot of, I think, movement. That the whole point of this, though, is to move in that direction, to have like a sort of a theory for this going forward.
Yeah, of course, what makes so one, just want to say, I was totally wrong about Tim Verbal. Oh. I think I was thinking of Tim Veblen.
Oh, okay.
Anyway, like Tim Verblin, I know because of Versus. Yeah. So he's like some leads at Versus.
Okay.
And so I think I just know him from a bunch of first papers. As opposed to the diffusion imaging experts at CMU.
Yeah.
It's there's still this very like CogSci. Yeah, the problem of scaling this to Anthrobot is, of course, they have no nervous system.
Right.
and I guess it just kind of pushes Michael Evans' point about like there are many diverse intelligences.
Right. Yeah.
Yeah. And
cognition all the way down.
It is, yeah. But it is, yeah. So that's certainly what makes it interesting, is that like you have to understand this as being some possible from some collection of cells that don't even have a nerve, don't even share a nervous system, let alone understanding how single cell behavior is is guided, and yeah. So great talk from I don't know what the Leverhulme is. It the Leverhulme Institute in Cambridge?
I think so.
It's something like that, like physics of life, or I forget what their mission is, but great talk on kind of like chemotaxis and ciliary behavior, and single cell Volvox, I think. Yeah. Some of those things just have trouble, some of these concepts I have trouble putting into a cellular box.
Right.
But I think that is more my problem than the single cells, or just like habits of language,
right.
make it such. Yeah. But I also noticed that there was a very small group of people publishing these papers.
Yeah.
So like some of those authors, I could see that the reviewers were like the same authors from the first paper, Ines was the reviewer of the one, and yeah. Anyway. I'm not suggesting something bad, but just that it's just kind of like it's a very small community.
Yeah. Well, anyways, I thought that. Oh, go ahead.
And the final thing that like when we were kind of thinking about Thousand Brains, like you go to the discussion section of that paper, and it's like the first reference is the Thousand Brains. It's like it's totally Hawkins 2017.
Yeah, there's a lot of self-reference and a lot of this stuff. I mean, some of it is that it's like people are starting to think about it, so you don't have a lot of deep background in it. But also, like, there's like who's going to review the paper, and that's a problem with peer review.
Absolutely. I mean, like there's like two MRI guys that are both resigned to the fact that every time they publish a paper, the other one gets asked to review it. Because they're like, nobody understands this stuff except these two. So at least there's two of them.
Yeah.
So you can at least call it peer review.
Yeah.
But yeah, it is funny. But it's well.
Okay. Yeah, I thought that was interesting.
It's also fun, just thinking a bit about me throwing stones at all the organoid people and bioprinting people that have got companies filing patents, and it's just like how many students of Thurston or people associated with Thurston are like working at robotics companies right now, like pushing active inference.
Yeah.
Because it's not just verses.
Yeah. Okay, so I did talk about in our last meeting. I did talk about Valentino Braitenberg and how he was a neuroanatomist and how that may have influenced it, or it did influence his work on vehicles and some of the some of the cross topics there. Wanted to follow up with some additional stuff on that. Mainly this idea that Valentino Braitenberg is what he called himself as a biocyberneticist. So we talked about the cyberneticists like Ray Walter, who did a lot of things with robotics, or Ashby, who did some things with intelligence. And then, we had people who maybe weren't as connected to that original Maele conference as, we would like, but they're still sort of in that cybernetics camp. So Braitenberg was kind of considered a biocyberneticist, which is applying the principles of cybernetics to biological systems, in this case also simulated systems. So I have some readings here that, these are kind of deep cuts. So they're some of them are old. Well, they're all old because if they're written by Valentino Braitenberg, they'd be, written maybe in the 20th century. But we'll see and then I have something to show in terms of how we think about some of these concepts with developmental Braitenberg vehicles and how his ideas square away with that. So the first paper is this Advanced Physiological Sciences article, and I'm not quite sure what year this was because they don't put it in the citation. But this is volume 16, Sensory Functions. This is Anatomical Basis for Divergence, Convergence, and Integration in the Cerebral Cortex. So as I mentioned before, he was a biocyberneticist. There's a biocybernetics wing of the Max Planck Institute. So they have this, they still actually have biocybernetics as an area of specialty. And I guess in this case, what they're doing is looking at mainly biological systems like the brain in this sort of cybernetics way. And I don't know what cybernetics means now, but this is what Valentino Braitenberg was thinking about. So what his concern was here was looking at the anatomical aspects of cerebral cortex, and then looking at some of these, the electrophysiology of it and the anatomy and how that comes together. So he talks about cortical electrophysiologists, and he says they're beginning to generate functional models which must eventually be fitted into the known structures of the cortex. So this was many years ago now. So I'm not sure if this is still true, but I guess we know have functional models now, but again, we have to fit this into the known structure of the cortex, and that can always be challenging. Quite independently from this ancillary role of histology, the structure of the cerebral cortex is impressive enough to invite speculation on its own. Particularly the view of the cortical computer, derived from the electrophysiology of sensorimotor areas, is quite different from that which the global view of the cortical anatomy inspires. So there's this idea of the cortical computer and how this is based on the electrophysiology of certain areas of cortex, and then there's this global view of cortical anatomy. These two views will eventually converge. As a counterweight to the prevailing trend, I shall collect here some obvious facts from cortical anatomy, which are frequently overlooked, although they might significantly contribute to our ideas on the working of the cortex as a whole. So he was struggling with this idea that there's this, there are these areas of cortex which we study quite intensely. We have computational models of those, and then we have cortex in general and what that is. So this is, again, kind of thinking about the neuroanatomy, but also thinking about the function, and then, kind of putting that together. So he kind of goes through the macroscopic anatomy. He goes through the description of the synapses. This is all descriptive, so this is not necessarily, it just tells you what's there. and then talking about some of the classification of synapses, and then the density of neurons, the types of neurons, relative number of the three types, axonal density. So it's like this kind of laundry list of what he's seeing in this model. This is, this is the albino house mouse. So this is being compared to human cortex, but focusing on the albino house mouse. So you go through all of this description, divergence and convergence. So this is interesting. If this is a learning machine, it is important that it be free to learn whatever patterns are presented. There must be the possibility of a connection from every neuron to as many as possible other neurons. If a pyramidal cell has 5,000 synaptic contacts, with how many different cells is it connected? So this is where we have one cell being connected to every other cell, having this fully connected architecture. And so there are two lines of arguments to suggest the answer is with almost as many, and this is cited in Braitenberg, 1978. First, we consider that the cross-section of the dendritic tree of the neuron can derive a geometric measure of the probability of a fiber entering its confines and hitting it once, twice, etc. The probability is very small for 2 or more hits. So I think this is a picture of this where you have this dendritic tree and you look at the architecture of the tree and see how many times it sort of intersects, and you're getting a measure of the sort of full connectivity of the cortex. So this is a cross-section of the basal dendritic tree of a pyramidal cell, as seen by a straight fiber entering its confines. So this just shows this, and then there's this probabilistic estimation that's made. What are the surface area of this region and the area of the circumscribed circle? The probability of hitting it twice is proportional to the small hatched area here. So these cross twice, and you see this here. Second, we may picture a neuron as a cloud of postsynaptic sites, intermingled with many thousands of clouds of postsynaptic sites of other neurons. If a fiber entering that region picks its contacts at random, we may calculate the probability of it hitting that neuron once or many more times by the binomial distribution. Here again, it turns out that the probability of multiple hits is quite small. So he's working out this problem by doing sort of a topological model and figuring out how, if you just generate connections, how often do they cross each other, and you can calculate that probability. And then doing a sort of a probability density function approach, where you just assume just a cloud of interactions. And in both cases, it turns out that this is quite a small probability of happening. So what does this mean? This means ultimately we derive the picture of the cortex as a device which reshuffles the incoming information in as many as possible ways, perhaps in order to discover correlated activity and to incorporate this knowledge in the form of what he calls cell assemblies, which of course are a thing that he proposes in 1978, cell assemblies in the cerebral cortex. And then, we have more modern work that kind of expounds on that. So that's the first paper. The second paper is this brain size and number of neurons, an exercise in synthetic neuroanatomy. This is more recent. This is from 2001. And so he kind of gives these different models of sort of invariances in comparative neuroanatomy. So let's go through the abstract. Certain remarkable invariances have long been known in comparative neuroanatomy, such as the proportionality between neuronal density and the inverse of the cubic root of brain volume, or that between the square root of brain weight and the cubic root of body weight. Very likely, these quantitative relations reflect some general principles of the architecture of neuronal networks. So we have these quantitative aspects. We have brain volume, brain weight, body weight. So this is the sort of allometric scaling that you'll see in biological systems. Brain growth is, a brain can grow, but it's usually with respect to the body. So you get bigger brains in bigger animals, smaller brains in smaller animals. And then that scaling can change itself. That scaling function can change its slope based on the expansion of cortical regions, or sometimes in birds, it's the telencephalon. And so you get this sort of scaled relationship, and it gives us these quantitative measures that we can get a sense of the architecture of neuronal networks. You also see this in the hippocampus of different organisms, especially if they engage in a lot of foraging versus not. Under the assumption that most of the brain volume is due to fibers, we propose 4 abstract models. So the first is constant fiber length per neuron. 2 is fiber lengths that are proportional to brain diameter. 3 is a complete set of connections between all neurons. And 4 is complete set of connections between compartments, each containing the square root of the total number of neurons. So these are 4 abstract models. These are just assuming different things about the brain volume and what's inside there. So we kind of think that most of the brain volume is due to fibers and connections packed in there. We also, can assume maybe a constant fiber length. So if we assume a constant fiber length, and we assume that most of the volume is due to fibers, we can then calculate the number of neurons. We can also assume the fiber length is proportionate to human brain diameter. We can assume a complete set of connections between all neurons, which gives us maybe a number for the number of connections and then the number of neurons. And then 4 is a complete set of connections between compartments, each containing the square root of the total number of neurons. So this is where we have modularity. We have these modules that are connected, selective with very few connections, and then each module is fully connected within. And then so talks about the models in terms of what they correspond to. Model 1 conforms well with the cerebellar cortex. Model 2 yields the observed comparative invariances between the number of neurons and brain size. And this is, of course, this allometric scaling idea. Model 3 is totally unrealistic. So that is our complete set of connections between all neurons. So that's actually totally unrealistic. And then model 4 is compatible with the volume of the hemisphere white substance and different from mammalian species. So what this paper is suggesting, kind of is maybe in contrast to the other paper, but also maybe supporting the other paper, is that we can think about the brain or the volume of cortex in terms of how it's connected. We can work backwards, find the number of neurons, find the connectivity patterns, and then, make certain assumptions about those connectivity patterns and see how well that model fits the data. So, no, we don't have fully connected neural networks. We actually have maybe this modular network where there's maybe local full connectivity and then selective global connectivity. And that would fit what we see in mammalian species. So he goes through, a lot about this sort of set of assumptions, how these are set up. We have these 4 models, and talking about real brains. Unashamedly unrealistic as our schemes of 1 to 4 may seem, they do yield some values that are remarkably close to measurements of real brains. And so we're going to go through some of these comparisons. And so, yeah, we know from experiments that allometric scaling is a 2/3 power law. The number of neurons and the 2/3 power of brain weight is something that was established experimentally back in the 1950s and 1960s. So this is where we have the allometric scaling for the number of neurons versus brain weight in grams. So the weight of the meat, or the weight of the sort of the tissue, versus the number of neurons within that volume of tissue. So you can see that, monkey, cat, rat, and mouse all fall along this slope. And then humans are out here as an outlier. And the reason humans are out as an outlier is because in a lot of primates, you have this expansion. In great apes, you have this expansion of the brain, and in humans, you have the greatest expansion of brain tissue. And so you have this outlier out here. Okay, so then, this just talks about like thinking about this approach of what he calls synthetic neuroanatomy, where we're using simulations, we're using mathematical models to model a lot of this stuff. And so I think it's a useful approach. It's definitely something that we've picked up on in our group. And this is a slide I made up of these 4 models of the brain. So in black is from this paper we just talked about. And then in red is sort of the way that this is dealt with in the developmental neurosimulation paradigm. And if you're interested in more about this, we have a series of papers on our website that have been published in various places that go over some of these concepts. And so developmental neurosimulation kind of addresses all 4 of these models in different ways. And so if you think about like a vehicle, because he's talking about real neuroanatomy, but eventually he gets to this sort of model of vehicles, and thinking about the vehicle versus the actual brain. We'll talk about vehicles here, but he's talking about sort of real brains. What we're interested in is using vehicles as a means to get to simulated brains or developmentally relevant brains. So then the first model then is brains of uniform density. And this is where we have a compartment which contains a brain. The density is uniform across that. The volume is sort of, there's a volume and a density of content. So there's this little container here, this gray region that has a certain density and within that density we have neurons and axons, dendrites, so we have the connections and the neurons. And in developmental neurosimulation, this is a size of a network that's growing. So in developmental neurosimulation, our network is constantly growing within a volume, or the volume is expanding, and this expands the size of our network. And so we kind of approach this in a little bit different way. Remember from our last discussion, Braitenberg kind of downplayed the significance of development, or at least he didn't talk about development in his work. In our case, we're explicitly interested in development. So we're interested in the size of this neural network and this network of connections. And for a uniform density, we probably have a fully connected network. And so we characterize our matrix of network connections as a determinant, which is here, A. And so what is the size of A? And so in developmental neurosimulation, you have this determinant that gets recalculated over time as the network grows. And so this grows in size. And this can be also stated in terms of density. I've not done this here, where you have a restricted space in which you can put more and more neurons. So the size, I should also probably put the density of A as well, but I'll do that later. The second part is this network here in this vehicle from sensor to effector, which we can characterize as Wij. And this is where the length of each of these connections is proportional to the diameter of the brain. So as Wij grows, as the body grows, this is in Braitenberg's work, this is brain-body scaling. In developmental neurosimulation, this is also brain-body scaling. And in this case, we don't have just a compartment of a brain. We have this mapping from sensor to effector, which as this distance between the sensor and effectors grow, or the relationship between them changes, we get a different length of our connections. So our connections are, we have things that are connected. So we have sensors connected to interneurons, connected to effectors. We can describe that with Wij, but we also get more connections. We also get a longer distance between the sensor, the interneuron and effector, and so forth. So we can characterize Wij both in terms of growth, in terms of number of units, but also in terms of the length of these connections. And of course, biologically speaking, the length of the connection is important because the longer the connection, the more conduction delay you get, and the more leakage you can get unless you have myelinated axons. So these are, things to consider. I don't know if we consider this in a synthetic model, but definitely in biology. Then models 3 and 4 are the first. Number 3 is a complete set of connections between neurons, which is just basically this mass of connections without any modularity. And then 4 is where you have this fully connected end to the half compartments. And these are modules with limited connections, but internally with complete connectivity. And so we can bridge those two models actually in the developmental neurosimulation paradigm by saying that a selective set of connections between neurons involves end to the half compartments. And so we can actually have these kind of compartments play the role of interneurons or other modules in an agent brain. Whether those are functional or not is, we haven't really gotten to the point where we would test those kinds of hypotheses. In any case, so let's go on to the next paper here. And this is from 1974. So he was doing his work in this, from the 50s to 2001. So this is like a long period of time where he's working. But this is from 1974. And again, he was doing a lot of neuroanatomy. So some of this is just really cut and dry neuroanatomy. It doesn't think about too much about the simulation aspects or the modeling aspects. But in the last paper, as we saw, there is this very strong simulation aspect, biocybernetic aspect. So, summary of this paper, he's working in the visual. So in the first visual ganglion of the fly, Musca domestica, there are many similar visual channels or cartridges connected to each other by various systems of fibers, the most regular of which consist of the collateral branches of the L4 neuron, which is contained in each cartridge. The 3 collaterals of L4 run to 3 different cartridges, one of which is the parent cartridge of the neuron, the other 2 being neighbors of that cartridge in 2 directions on a hexagonal array. Within each cartridge, there are thus the endings of 3 collaterals from 3 different L4 neurons. These make close physical contact, and serial sectioning shows that each collateral is presynaptic to the other 2. It follows that there are reciprocal synapses between any pair of these collaterals. And then the network of relationships thus set up is the simplest scheme of coupling in a hexagonal array. So he shows images of this. So he works in this area where he's thinking about flies and their visual ganglion. So in dipterians, the visual field is resolved in discrete points, each corresponding to the central axis of one ommatidium. So the fly visual system is different from the mammalian visual system, in that they have these small ommatidia that form discrete points or dots in a visual array. So, we have sort of this, call it a camera-like eye, which has like spatial resolution, more continuous spatial resolution. We have, we can detect shapes and blobs and things like that. In the fly visual system, we have these dots, and you can present things in terms of dots, and they find patterns between dots. And so this divides things up into these subunits, and there are thousands of subunits in the eye. They function much differently than our retina. So this is, kind of the basis for a different type of visual system, and then looking at how that's organized. So this is the arrangement of L4 collaterals between the L4 main fiber, which is the black dot, and the cartridges, the white circle. So you have this sort of organization. It's a different type of organization than the mammalian visual system. And I think this is important because a lot of what we do in pattern recognition in our studies of visual perception, say using a neural network, is to assume this sort of model that is presumptive of mammalian cortex. And this is not mammalian cortex. This is a very different way of organizing a visual system. And so this is just showing how these, how these systems are organized, how they're wired up, and then how they function. So this is an electron micrograph of a synapse. So this is an electron micrograph where you see the different structures. They're pretty well resolved. And he shows he's labeled L3, L4, R6. And the receptor cell axon is R6. The neurons are L3 and L4. And you can see the connections here. L1 and L2 are over here. So he gets through the neuroanatomy, he talks about the connectivity. He has this diagram of reciprocal contacts between L3, L4 end feet. And so this just shows what the cartridges look like. So remember, this is all neuroanatomy, and one of the bases for Braitenberg vehicles was to say, okay, there's this very highly specific anatomical relationship between sensor and effector. If we look at an insect, we can see that there are these structures in the sensor. We have these structures in between the sensor and effector, which is kind of the brain wiring, and then the effector, which has its own structure. And so what he's doing is working out all this. The idea is that if you work out the anatomy enough, you can get this very precise model that can produce all sorts of behaviors, which is what he was interested in the book Vehicles. And so this just really kind of describes this sort of system. Our findings indicate that reciprocal synapses, as described in Rawl et al. 1966 in the olfactory bulb of the rabbit, may be a more widespread feature of nervous systems than has hitherto been supposed. So his argument also is that some of these aspects of neuroanatomy, of wiring, are universal across different nervous systems. So we should lean into those universal aspects. So yeah, he talks quite a bit here about symmetry and about some of these synaptic relationships and how they might be, really kind of, if we can characterize them well enough, we should be able to characterize the behavior they produce. He also has this other paper from 1974, Thoughts on Cerebral Cortex, talking about his thoughts on cortex. So this is kind of where he thinks about it as an input-output system, much in the spirit of cybernetics. So input-output means you have a sensory input, you have some sort of output, and then that gives you behavior. So I'll just read the abstract here. So the cortex is often described as a network processing information in the direction from sensory to motor areas. So going from sensors to effectors, the motor effectors, the sensors that are bringing in stuff from the environment. That requires an input of sensory information, output of motor behavior, and something in between. So this is kind of the assumption. However, the structure of the cortex is asymmetrical only in the vertical direction, suggesting an input-output transformation between layers rather than between areas. So if you think about the brain as having these modules or having these layers, the asymmetry is only in the vertical direction. This operation must be a very generally applicable one, since the plan of the cortex is basically the same everywhere. So in an attempt to understand it, a skeleton cortex of only pyramidal neurons is considered. They are characterized by a double dendritic expansion, an apical one in which the first layer, which is considered as the input layer, and a basal one which receives excitation from the axon collaterals of other pyramidal cells. So when he talks about layers, he's talking about the layers of cortex going from 1 down to 6 and kind of coming back up. that's what he's kind of getting at with this. If pyramidal cells learn, perhaps by growing dendritic spines to respond to frequent constellations of activity in their afferents, each will learn a property of the input through its apical dendrites, provided it was preceded by other properties sensed by neighboring pyramidal cells, which influence it through basal dendrites. So in these layers of cortex, you have a lot of feedback, but you also have this functional specificity for each layer. Thus, the pyramidal cells will code its input in terms of properties, which have a tendency to follow each other. This will be a coding which reflects the causal structure of the world. Various uses of a network embodying the conditional probabilities of events in the input is described, including the recognition of familiar sequences and prediction. The local variation of fiber patterns in the cerebral cortex, described as Maele architectures, is interpreted as a macroscopical expression of the different statistics of the set of conditional probabilities linking events represented by individual pyramidal cells in different areas and in different functional contexts. So this is going through his thoughts about the sort of the vertical asymmetries of cerebral cortex, much more than between regions, and how that plays. So he's really kind of mapping out this sort of theory of cortex. And this is not something that the Numenta people talk about. They focus a lot on Vernon Mountcastle's work on discovering these kind of layers of cortex and their kind of relationships between one another, thinking about things in these cortical columns, which is just a stack of these different layers of cortex arranged in units that do things like process images or process sound, and kind of going through each layer, doing different aspects of the processing. And so, he gives this conventional view of cortex as sort of these modules that are distinct sensory areas. So you have connections between, say, sensorimotor and motor. You might have visual and auditory, and you have to move between the regions. And so this is kind of these regions of cortex, and then you have the symmetry of the cortex, which is this layered cortex. So he says the anatomical picture does not bear out this sort of model of distinct regions. If I am given a small cube excised from a logical network showing the same asymmetrical pattern on 4 of its 6 sides, and an isotropic organization on the other 2, I shall hardly suppose that the network processes information in the direction indicated by the arrow A parallel to the layers of the asymmetrical organization in B. So as you move across the surface of the cortex, that's a different thing than moving up and down the cortex. That's kind of the point he's making. Okay, and so just thinking about the anatomy of the cortex gives us sort of, you have to reinterpret that in this differential view. So, he's talking about this in 1974, and actually he's modeling the cortex using the skeleton cortex, where he's kind of thinking about it, just stripping down everything in the cortex to these pyramidal cells. And again, this is an early version of computer simulation, or, thinking about this in terms of a computer simulation. And so this is a basic network of pyramidal neurons or pi neurons showing the afferent fibers from the first layer, apical dendrites, basal dendrites, and so forth. So these are the apical dendrites, these are the basal dendrites. So this is it's a pretty good paper. This actually reflects a little bit on what pyramidal neurons represent. So each pyramidal neuron is connected in its own special way to a particular set of fibers in the input layer through the branches of its apical dendrite. We may say that each pi neuron represents a property of the input in the sense of Klein's 1956 representation of events in nerve nets, namely in the sense that the pi neuron becomes activated when an event has occurred in the input that is the property of which the pi neuron stands for. We disregard, for the moment, the important question of how much synaptic influence onto the peripheral portion of the apical dendrite will ultimately arrive at the initial segment of the axon. But we can model this anyways. So he builds this nice model and uses pi neurons as memory elements as well. He thinks about this in terms of the engram and in terms of different types of associative learning and how that relates to modifiable synapses. So he's really thinking of this at the anatomical layer, but he has interests in, traditional cognitive questions like memory, associative learning, and so forth, sensorimotor learning. And so the assumption of this kind of memory in the pi neurons throws new light on the question of what they represent. Having learned in the fashion described, they represent properties of the input which have a tendency to occur in a certain sequence. So he's thinking about sequential learning or sequential information. Or if emphasis is not as much on the temporal order of events as on the fact that occurrence of one event raises the probability of the occurrence of another, such as in Hebbian learning. We might say that pi neurons represent those properties of the input in terms of which the causal structure of the world represented becomes evident. If a connection axon collateral-based dendrite has been established as a synapse because it has frequently contributed to the state of excitation of the neuron, it follows that the synapse is a record of the conditional probability linking the activity of the presynaptic neuron with that of the postsynaptic one. So in terms of the outside world, that synapse stands for a causal relation between the events represented by the two neurons. So he's not just suggesting there's associative learning, but that there's this causal representation. So, that's something that may be controversial even today to think about this in terms of a causal relation, because there are a lot of feedbacks as well. And, that's something that I don't know if he modeled that, but that's something in the brain that you often have. And knowing, like, there's this whole area called credit assignment where you're trying to figure out, like, what neuron is contributing or what set of neurons is contributing to what behavior. This last paper are two views of the cerebral cortex. This is from Brain Theory, 1986. And this kind of talks about cerebral cortex again. And he talks about reciprocal positive feedback. And so he makes this comment here. Cortical anatomists and physiologists learn to shape their findings so as to make them acceptable to the theoreticians. The resulting situation of reciprocal positive feedback has some stable solutions. So there are a couple, let's see, how many are there? There's 5 solutions here. The first is random networks with and without learning. Lashley's philosophy is of this category, as is Hebb's theory of cell assemblies. And so Hebb also had this theory of cell assemblies. Rosenblatt's perceptrons is also a descendant. 2, the circuit diagram is in the spirit of radio engineering. The amplifier entered neurophysiology from communication engineering. With it came various ideas. So this is kind of focusing on this aspect of cybernetics as a circuit diagram. 3, the digital computer and a logical theory of nerve nets. This was soon recognized as a misleading analogy that the digital computer has, at any rate among all models of cortical function, the unique distinction of being a very useful machine. And the theory formulated by McCullough and Pitts, made more palatable by Klein, lent the brain a flareable mightiness for which was gratefully recognized by many. 4, the idea of projection derived from optics. So this is where you get these maps of the body's surface or some sensory space repeated many times in cortex or in some cortical structures. So again, projectionist research is still flourishing, but we don't really understand the details. I think we're better off now than we were then, but still, this is sort of derived from optics, where you're projecting things in different ways through a lens. In this case, the lens is through the nervous system and the processing of the sensory organs. Number 5, the idea of localization of function, derived from dualist philosophy. If you are utterly convinced that the connection between mind and brain escapes us as a matter of principle, all you can do is find out what part of the brain is related to what faculty of the mind without asking any further questions about the nature of the connection. This leads to very useful partitioning of the brain and perhaps less useful dissection of the psyche on anatomical grounds. So he kind of goes through these points, and then in view of these findings, it would seem difficult to sustain the essentially random network philosophy, which has grown dear to our hearts, on the basis of many anatomical facts. However, I will show that it is possible to make the 2 points of view quite compatible with one another. So he goes through this, talking about the neural pill in the mouse cortex. So he says that all the essential features of cerebral cortex can be found in the mouse, except that in humans it's scaled up by a factor of 1,000 in terms of size. It is a task requiring some experience to tell a histological section of a mouse cortex from a human one. Before we approach the problem of the shape of neurons in the cortex, we take a more global view and collect some quantitative data. So that's what he's doing here. Making some measurements, the density of neurons, distinctions between synapse types, axonal density, and then the axonal trees of various neuron types, dendrites, the cross section of a dendritic tree. So there are a lot of measurements you can use from the neuroanatomy. In principle, these figures provide the basis for a connectivity matrix of the cortical neurons, but of course the form factors reflecting the shape of dendritic and axonal ramifications are also involved. The probability of a connection between any two neurons in a block of tissue depends crucially on their distance. There are 2 ways of calculating this for neighboring pyramidal cells. One is based on the assumption that the connection via straight axon collaterals and that the form factor on the receiving side is described by the cross-section of the dendritic tree. So you can describe the shape through these metrics that he's collecting from the neuroanatomy and then model these as appropriate. So, what are the propositions which can be extracted from these anatomical facts? 4 of them are presented in Table 3. So we have the first one, which is A, and they involve certain measures, and that is most synapses are on spines. The second, which involves these other measures, most contacts are pyramidal to pyramidal. The third point is that the number of neurons afferent to a given pi cell is almost as great, around 5,000, as the number of its afferent synapses. Similarly for efferent synapses. So afferent are the ones heading towards a cell, efferent are the ones coming from a cell. And it's just a way, in this case, it's a neuroanatomical way to describe feed forward and feedback. Then D, which has another set of measures still. The cortex is a mixing machine. The activity is relayed in ever new combinations from one set of neurons to the next. So you have this idea, or this idea that was prevalent, I guess, early on, that the cortex is this all-to-all connectivity machine. But then, we kind of thought, well, maybe not, because there's, distinct function. And so, there's this idea of modularity. There's also this idea of a mixing machine, where there are new combinations generated all the time. And so, this kind of, I guess, is maybe the inspiration for connectionism, but it gives us this sort of model of, connections versus function. The upshot is proposition D. The cortex is a system of fairly uniform units, pyramidal cells, with a smaller number of neurons for different kinds of interspersed, connected to each other by widespread but very weak links. So weak, in fact, that the activity of one single cell can hardly exert an appreciable influence on the others. We must assume that the elementary event is constituted by fairly large sets of active pyramidal cells, so that fresh sets will be activated in succession through the synapses preformed in the cortical tissue and molded by a learning process. The picture I have in mind is influenced by Hebb's theory of cell assemblies. by Abalis's Sinfire chains by various developments in this work by Palm. So I guess in this part of his career, he was really kind of thinking very theoretically in terms of like, what is the neuroanatomy I'm looking at mean? Eventually, of course, he'd go on to write the vehicles book where he put all this together in these toy models called vehicles and kind of like stating out some of these, like cleaning up some of these observations and stating them as the vehicle models. So here's a wiring diagram. Here, illustration of a naive neural model explaining some of the effects described by Hough and Wiesel, who did the cortical columns processing basic shapes in visual cortex. So in A here, the array of dots represents the sampling points in a portion of the visual field corresponding to individual cones of the retina. So you have this visual field, you have the cones, they're dots. They're not like Ommatidia, like we saw with the insects, but they do have this sort of spatial array that they're located in. And they represent different parts of the visual field. So this kind of shows how they phase shift, and then these two in C, it shows the connectivity between these different parts of the visual field as you rotate them 18 degrees. So these are things that belong to different orientation columns. So one orientation column represents the visual field like this, and the other one represents it like this. They're shifted in orientation, in angle, but they represent the same spatial information. Then he has some nice drawings of orientation specificity. This is an array of hypercolumns seen from the top. So he's really into drawings and linking that with these histological images. Okay, so I think that's it for the Braitenberg stuff. I went through and did a deep dive on that to get a sense of what was out there in his work and his legacy. And I think it's really interesting. So I have the one that we did last time in the last meeting, and then this one. And I hope that's, kind of inspiring for people to think about, how to apply that to agents and other things like that.
It's just a great review of kind of the range of his work. And, his interest in connecting this. I mean, not only finding common structures across different species and types, but relating those to computational structures, paradigms.
Yeah.
I mean, again, sort of what you see coming together in vehicles.
Right.
But
yeah, this is like about 10 years maybe before vehicles leading up to it. So he's thinking like kind of, he's of course doing his neuroanatomy early in his career, and then he's moving towards this theoretical thing, this theoretical interpretation, and then moving towards the vehicles, which includes some of the things, but some of, he's not focusing on cortex, which is interesting because the cortex is a major part of this work, but doesn't show up in vehicles. Which begs the question, like, what is the relevance of cortex to vehicles or the cortical models to vehicles? Yeah. So again, yeah, this is all, and then relating it back to the last thing I talked about, which was, of course, there was a lot of free energy principle, but there was some work that was Mementa flavored with cortical columns, and that's interesting too because cortical columns are this organization. It's different from the sort of the connectionist view where you have this, you have layers, but the layers are sort of, they don't have a structure, kind of like these random sets of connections, and they process things. So it's kind of like maybe an abstraction of the cortex, the cortical layers, but it could also be like a sheet of neurons. It's not clear exactly what that analogy is. And it does things, it produces interesting things, but what's the anatomical rooting of that? With the cortical columns concept, it's very explicit in terms of the anatomy rather than being vague or implicit. And so that means that opens up this sort of expectation that it can model like sensory processing or multi-sensory processing better. And I don't know if that's true, you know.
Yeah.
Yeah. So yeah, so I don't know if Jesse had anything to say or if he's available.
Hey,
Hi.
I kind of came in a bit late, but I had happened to see on YouTube that you made like the Braitenberg vehicles deep dive or something like that, and then I look forward to going back to either seeing this recording or looking on YouTube too. I think it's I've kind of um, I've been hoping for or anticipating or wanting to see something like this because I feel like it's such an interesting tie to many things in the lab. So, excuse me. I look forward to going to that again. I don't know what was covered earlier today. I might share just a couple things. I think we're kind of at the end. Yeah, it's past. I won't say a whole lot now.
All right.
Anything that happened earlier that you think I should particularly go back and look at or pay attention to or updates or things that were shared?
I mean, Morgan gave a nice extended view of the stuff that he talked about yesterday in the open source meeting on bioprinting and some of the stuff they're doing with cell culture. Then I went over a special issue of it was on BioNeuroAI, and it was a special issue with Adam Safran um, and colleagues. They were doing a bunch of things with respect to enactivism and free energy principle and other topics in the space of robots for robotics.
It would probably be good, and I think it might be up. Let me two things of note this week on those similar topics would be NeuroMatch has started, and I'm just a project mentor, so I'm kind of not super involved.
Yeah.
But that has started, and then apparently I don't even I don't even remember which I signed up for, but either way, I'm in both. I'm a mentor for the NeuroAI and the mentor for the other one.
Oh, okay.
So that's exciting, and it's NeuroMatch is always very intense, and it's kind of nice to be involved, but also not too involved this time. Because there's so much other stuff going on, and it's actually as I mentioned in previous Friday meeting, it's super duper there's a lot going on. What I want to do, what I'm going to be doing this weekend is, yes, finishing the finishing the presentation. That's that's one thing that I can certainly do and want to do. But more so than that, I can't find my past events for this thing. Oh, this is disappointing. I was trying to show something from Echolapta this week that's super relevant.
Oh, okay.
Because we had, yeah, here we go. All right, finally, took forever to find, but here it is. I'll just very quickly mention this. I forget if we've talked about who about this particular person before. Let me go to it. All right. Have we mentioned Anna? Anna Cianica?
Oh yeah.
Embodiment.
Yeah, embodiment.
Okay. It was long awaited. Finally getting this up. This happened this week. It was Elan Baranholtz, who is very interesting in the sense of he's doing a lot in the space of LLMs are, I think, the AI hype version of what he's saying. Oh yeah, kind of like LLMs are how our minds are working. But that's maybe a bit unfair, and I don't think he'd even agree with that. But a lot of Elon's doing a lot right now with LLMs and the structure of language and how language is processed in general, and relating that to cognitive science and AI stuff. And Anna is much more biological embodiment focused. And it was interesting because the discussion, which I don't think is online yet, I stumbled around thinking it was on YouTube, but I don't think it is yet. The discussion was very interesting because Anna's a very interesting. She enjoys being a philosopher, enjoys exploring, was very knowledgeable about embodiment and kind of the Varelian lineage. So sort of things that we've talked about, and it's interesting to kind of tease apart where the defenses of embodiment, which I'm sympathetic to, and what Elon's doing, which I'm still trying to sort out exactly how I feel about it, because part of it reminds me of the brain, the mind is a blank, like that concept, and okay, now LM's the latest version of that, which is a paper that I'm supposed to finish. But also, there's if I had infinite time this weekend, which I don't think I will, or various other things, I would assemble. I would finish that paper, and then I would also assemble this sort of one of the most interesting things coming up in ecological discussions about mind stuff is like I'm kind of putting forward this term. I kind of want to make something using this term, so it's there. But basically, one of the main concepts of language is coordination, or coordinating, like something really coordinating ideas, coordinating like relations to affordances, or in a pithy, quippy way, language as teleological coordination. So about goals. And that's I have a draft of something I want to write about that. Not because I really believe it as the best way to approach this, but as a way to try to, I think that lens would be really useful for differentiating what we were talking about in some recent conversations there. And for those who don't know, Ecolapto is sort of it's sort of like an impromptu. Some things are very planned out. Some of them are much less so. But this is sort of we have an opportunity to get Elon and Anna together. It kind of kept briefly, and so I was able to join the discussion, or sort of impromptu, but not quite. And we joined the discussion and talked about stuff, and it was good. But essentially, Elon is sorting out this LLM language implication. Anna is was a really interesting embodiment individual to talk to about a variety of things. And Addy is sort of working from a higher level on Addy, sort of Ecoloptus, I don't know, chairman, founder, lead, is working on essentially olfactory language, but olfactory language as an example of making languages or dealing with languages of different sort of forms or embodiments or affordances or sensory makeups. And just so it's a means to explore the space in general. And that's a very curious thing that's happening. And I you know, I'll leave it at that. When that's available, I might have 3, I think, and stuff like that. But that happened this week, and I think there was one or similar other event in that nature. But you mentioned, oh yeah, NeuroMesh stuff, and then the last sort of general thing I'll say is I want to check out the Adam Saffron stuff, and this as I mentioned yesterday, this is a huge week for a lot of the nitty gritty stuff, like a very particular take on professional development. I'm doing the micro MBA. I'm actively meeting with a lot of folks in the how you build certain things sense, and I'm excited about that for what I want to do with JoPro at large, but also some of my own personal projects, and trying to distill for people going into this space, what do they need to know? I think the term was like organizational literacy, and I that's that excuse me, I think is sort of adjacent to or embodies to me this sort of broader curriculum that fits the presentation of like how do you present to different audiences. That's sort of a very you're an agent or a node communicating in between some of these groups, and then how do we do some of the structuring? How do we do sure funding and all that other stuff, but like there's a lot of team in strategy, planning, communication that I think overlaps with the things we've been talking about. And I'm going to try to continue to concretize, to make concrete some of these things ahead over the summer. And so more on that ahead.
Right, that sounds great. Yeah. Well, thank you, Jesse, for that update, and thank you, Morgan, for your update. And why don't we call it a day? Thanks for attending.
Yeah, I might reach out to you folks both about some of the discussion series things we've been talking about, but that's about it. Otherwise, have a good weekend, everybody.
You too. Thank you. Bye.